Hello, welcome students. In the last episodes, we have studied about the prophase and metaphase type of mitosis cell division. Understand? And you know that mitosis cell division that takes place in the somatic cells first. In that, the mother cells and the daughter cells have same number of chromosomes. That's why it is called mitotic or equational division, because they both contain same number of chromosomes, that is mother cell and daughter cells. This division is also known as equational division. And next, this reproductions, in that one cell divides into two daughter cells. So mainly point, this takes place in somatic cells. Second, the number of chromosomes in both mother cells and daughter cells are same. Third, one cell divides into two daughter cells. That is the main features of mitosis cell division, which we have studied in the last video. And prophase, you know that in the prophase, this is the parts of a nucleus division, that is karyokinesis. Karyokinesis divides into four phases. One is prophase, next one metaphase, third one Anaphase, and fourth one Telophase. In the prophase, the chromosomes accumulate, they become distinct. In the metaphase, they come in between the spindle at equator. So we have studied that. Now, cell cycle and cell division. Now, mitotic cell divisions. In that we have studied prophase, metaphase. These two stages we have studied in the last video, but today we shall discuss Anaphase and Telophase. This division we shall discuss. So, first we will discuss the Anaphase. In the metaphase, we have seen that this is the phase of metaphase. We have studied in the last video. Now, the anaphase is that the spindle which you see, understand, the constriction takes place in the spindle, and due to that the chromosome splits in two halves. So there is a constriction in the spindle towards the pole, means the spindle becomes shortening, and that's why the chromosomes will split and the chromatids will go towards their poles. And you know that here energy is applied to separate these sister chromatids, and that ATP is about 30 ATP per chromatid. So about 30 ATP is needed to separate them from the chromatids are separated from each other. The energy is applied or energy is required, and that is about 30 ATP. Now this is so thus one most important thing is that you know that in the S phase the DNA amount becomes double. That DNA is divided in this phase. So DNA is divided equally in both sides. The DNA is divided equally in both sides, means one towards the half towards this side and half towards this side. So it is generally asked if DNA is divided in which stage. That is the anaphase stage. That is anaphase stage. The DNA is divided. Understand? And so it is it is also a short phase, but it is important because DNA is divided in that. Now the next phase is the telophase. Now the next phase is the telophase. In that telophase, this is the cell membrane. The chromosomes will go to their ball. Means first a spindle lost and chromatids gather at the pole. Then that is they gathered at the pole. Understands? Second, nuclear membrane appear. The nuclear membrane appears. The centrosome also takes their place, and all the contents are also divided, and centrioles is also a central nucleolus is also appeared. So centrosome, nucleolus appear, and so in this way one cell contains two nuclei, means two nuclei condition formed. Understand? And so the next stage, that stage is the completion of the karyokinesis, means division of nucleus is completed. Now the important one, that is cytokinesis, that is the one cell divides into two, means this is the conditions of that. Now the cytokinesis will take place, means for that there is a constriction takes place in that. The constriction takes place in that, and that constriction becomes deeper. That constriction becomes deeper and meet to each other. Understand? And finally, two different cells, two daughter cells are formed. Finally, two daughter cells are formed. Understand? So in this way, after the karyokinesis, after the division of the nucleus, the cytokinesis takes place. And in plant cell, the cytokinesis takes place by the formation of cell plate in between two nuclei. Understand? So that is the difference between plant cell and animal cells. That there is cell plate. In the plant cell, cell plate is formed between two nuclei to separate them. But in the animal cells, the plasma membranes constrict towards inside, and that becomes deeper, and finally two cells are divided. So in this way, one cell divides into two, that is mitotic cell divisions, and both the daughter cells have same number of chromosomes. Now the significance of mitosis. Significance of mitosis cell divisions. We know that number of chromosomes is same in both daughter cell and the mother cells in mitosis. First. Second, by this the number of cells increases. By this means number of cells. By this number of cells increase, and that's why it is responsible for growth. Understand? It is responsible for growth in the plants. You know, in the meristematic parts, these mitotic cells are the cells which are found, they undergoes mitosis cell divisions, and due to that the growth takes place in plants. So it is responsible for growth in plants and animals, that in all livings, by the mitotic cell divisions. And second, the self repair. If there is any cut and become wound, and that deep part is again swell up or again completed by the cell divisions. यानी जब कहीं कट के गड्ढा हो जाता है तो वो गड्ढा फुलफिल होने का काम, that is due to cell division. So they are the most responsible for the growth of the parts, growth of the organs. Understand? So any type of growth which takes place in the livings, that is due to mitosis cell divisions. And any repairing in the cells, that is also due to mitosis cell division. So these are the mainly important parts of the significance of the mitosis cell division.